So let me share my screen. Okay, so what I will be talking about today are a few projects that we've been working on with collaborators around the place for a few years now. And first I will thank all my collaborators deeply for the opportunities to do this work together. So what we'll be addressing chiefly is the idea that that one continues to learn and maintain skills across one's lifetime. So this is my little son in Woods Hole. He took the time when we were in Woods Hole over the summer to learn to play tennis. So he's not the primary focus of this motor skill learning talk. It's really his mother. So he needed a partner when he was playing tennis. And I used to play when I was in college, but hadn't played for many years. So I had to kind of dredge back those skills and figure out, remember, right, how to play. And so one can do that, right? You even years later, if you've learned a motor skill, that skill can stay with you and you can recover it and be reasonable. At some point, of course, he got way too good for me and we don't play together anymore. But, there was a moment that it worked. So, we'd like to know, how does one learn to produce precise motor behaviors, but also very importantly, as an issue of practice and performance, how you maintain that skill through practice. And at the time that you're playing, an important game, how do you sort of squeeze down the degree of variability that you need in order to advance your skills and play a very good game. So, obviously someone like Serena Williams is a whole other level, and she isn't a great example of someone that has come back to the court after major changes, after aging, after having a kid, and yet is still playing kind of at the top of the field. All right, so birdsong is often been thought about as a great model for motor learning. That's because birds, at least some birds, produce a highly stereotyped behavior, which means it's very quantifiable. So we can characterize the variability from trial to trial. The bird brain has a very well characterized neuroanatomy that serves as the substrate for this motor learning. Some birds, this one in particular, zebra finch, is a trial and error learner. It practices a song over and over again and improves over time. It retains some plasticity throughout life. It's able to learn as an adult, even though the song is largely crystallized at that point. And so this as a case where one might hope to connect kind of high-level learning algorithms deeply with the sort of biological substrate in terms of the neural circuitry, the neural cell types, the biophysics that underlie these learning models. So as most of you know, that birds, songbirds learn their songs in two phases. There's a memorization period where the bird listens to a tutor and primarily just listens. After time, it starts to kind of babble, practices the song, and then after about 100 days, the song crystallizes, and then the bird retains that song throughout adulthood. But it really is a trial and error process. So every dot here is a different song. This is characterizing how similar any one rendition of a song is to the final song model. And you can see that from day to day, right? The bird sings a wide variety of different songs, but that over time during this learning process, that song approaches its final target and the variability decreases. Nonetheless, as I mentioned before, the zebra finch are able to continue to vary their song throughout adulthood. And so a lovely sequence of experiments by Phi Lab and Brainerd Lab showed that in the 2000s, they forced a bird to change a particular pitch of one of the syllables of the song. So focusing on here, you can see my cursor, I hope. So it's focusing on this target region that has a relatively clean spectral shape. If you characterize the fundamental pitch of that syllable, and now you give the bird negative feedback whenever that syllable varies in a direction that you want to punish, right? You can force the bird to kind of gradually shift that syllable to a new target. Right, so here that negative feedback is given through a microphone that's attached to the skull. When the bird sings, at a pitch that's, say, too high, then that microphone will give a kind of that gives negative feedback. And so here, what you can see is that the bird is singing the syllable at different pitches, and now that you turn on the negative feedback, you can kind of push that syllable around from day to day and train the bird to sing at a variety of new pitches. All right, so let's do a quick dive into the anatomy of the song system, because we're going to be looking at the behavior and the wiring of a few of these different regions. So simplified version here, there's a pathway which is responsible for song production, area HVC, which we'll talk a lot about, and generates a very regular firing pattern that drives a nucleus RA that then drives the syrinx via the eighth nerve. Outputs from HVC also pass through these blue areas that collectively is called the anterior forebrain pathway. And these regions are necessary for learning and also for song maintenance. The VTA is analogous to mammalian VTA that produces dopamine, which largely innervates area X. Now, this architecture long ago has been sort of mapped onto the components of a reinforcement learning paradigm. So you think about the cortical motor pathway, which does song production. I'm going to be using this cartoon a lot, so hopefully that will be a little bit easier to parse. So song production pathway, or we'll always do in red, anterior forebrain pathway nuclei responsible for learning and memory. And the VTA is thought to be, as in mammals, kind of the source of the judgment or the value and reward. So one can kind of map these approximately onto this is the actor, right? This generates the behavior. Diane Sinowski introduced the idea that one should think about the anterior forebrain pathway as essentially an experimenter, that it injects variability into the song, and that allows this actor to learn, right? By experiencing fluctuations that then improve the song. One can then reinforce the fluctuations that are positive, that make a good change toward the song. And the VTA plays the role of the critic. So general questions that my lab and the field in general have been thinking about is, how does HVC generate robustly the sequence that serves as kind of the scaffolding for song timing? What are reasonable models of learning? HVC has to learn to wire up to RA in order to generate the output of the song. So how does that exactly work? What are the origins and modulation of variability that come from the anterior forebrain pathway? And finally, another key question is, what's the information content of this dopaminergic feedback? What is that actually telling the anterior forebrain pathway, and how does that help to shape song? So today I'm going to focus on just a piece of this, that these two components, so models of learning, and also the question of robust sequence generation and maintenance. Okay, so to show you, I'm sure many of you are familiar with this, but Michael Fee's lab showed years ago that HVC fires in a rather special way. So an individual neuron from HVC generally will fire once throughout a song, and with very precise timing relative to the song. So here's individual neurons whose timing sort of tiles the time throughout the song. Then RA neurons, because they're driving muscles, they of course will fire multiple times throughout the song, because each of those is sort of responsible for sort of activating a muscle that's generating these syllables of the song. Right, so the goal of a learning model is to figure out how to transform these moments in time into the action that the bird needs to do at that time. So we're really going to focus on this piece, on the HVC to RA synapses, and then the role of L-man as outputting variability into that pathway. So there's a classic model from Ila Feet's lab with Sebastian Sung, where L-man was basically characterized as sort of a random number generator, that it was responsible for injecting sort of random fluctuations into this HVC to RA pathway. and that they postulated that there was an action of a sort of a critic that was able to take the variations that occurred at each of these synapses, apply to them an evaluation of how positive or negative those were for the song, and then to strengthen the synapses accordingly. So if we zoom in here, each of these neurons in RA is receiving synapses from HVC, they're plastic, and that they are reinforced or not according to fluctuations that are produced from Elman. So we took that model and we used it to explore some findings from Tim Gardner's lab. So here's our version of that cartoon. We have HVC driving RA, gets input from Elman, as we know from the anatomy. That generates a motor output. And so we're going to assume that every, here we just look at two of the different motor neurons, they have to generate some temporal pattern, and that's the task of the motor output. So one thing that we know about birdsong is that it's remarkably stable. If you record songs over here, hundreds of days, 400 days, then the spectral signatures of those songs are extremely similar from day to day, modulo this small amount of variability. Tim Gardner's lab did some experiments where they used optical imaging to record from HVC, again, over hundreds of days. And what they saw was that, surprisingly, the neurons that are generating the song, right, these HVC neurons, here you see that they are firing precisely, but they saw that there was a small amount of variability from day to day. Some neurons would fade out, other ones would come in. And so at the level about 5%, there is some turnover in the firing times of the neurons within HVC. And so what we wanted to know was what implications does that sort of low level of continuous variability have for the learning of the network. And so we characterize that by thinking about, here's our HVC neurons firing at particular times. And then we introduced a small degree of perturbation, whereby here, for example, neuron 3 would stop firing for a certain amount of time. And we trained the network to learn under this degree of perturbation. And then we varied the rate of perturbation, so how often these perturbations would occur. And so without perturbation, if we just let the network wire up, let the HVC to RA synapses wire up, and you look at the error, then you get this red curve, right? So you quite rapidly learn a relatively low error song, and that error is maintained. If you introduce the small number of perturbations, then obviously, as you introduce that perturbation, the song will get worse, but then it will recover. And here it doesn't quite recover to the same level. So, we keep introducing these perturbations, and it never quite gets to that lowest ever error level that the unperturbed song gets to. So what we can now do is look at how that error changes, right, as a function of how often one introduces a perturbation. And the more often you introduce perturbations, you do take a slight hit, right, like a 5% or so, or less hit in the quality of the final song. And so why would the bird allow this degree of perturbation? Well, it turns out that there are real advantages to training the network this way. So here's one. If one has learned this original song, so the motor output here, drawn in black, was the original target song. If we now ask the bird instead to generate a slightly different motor output, so one could imagine that the muscles have changed in their qualities as the bird gets older, and so it needs to drive them slightly differently. So how well does our network learn this perturbed song? So what we find is that as one trains the network with more perturbations, it becomes able to relearn that new song more rapidly, right? So here we've got networks trained with again, different numbers of perturbations, and we look at the time to relearn the new song, and you see that time decreases as with the number of perturbations with which the song has been trained. And so what we have is a network that's more readily able to adjust to changes in the song. Another advantage is that it is more robust to damage in RA. So if we now kill off some neurons in RA by ablating them, and we ask what happens to the song in the presence of this kind of damage, we also find that the error from cell loss is considerably less the more perturbations that we train the network with. Right, so here's the error from cell loss. If it's trained with no perturbation, it's a large error, and that error gets smaller and smaller the more frequently we have perturbed the network during training. So why is this? What's happened in the network? So if we look at the connectivity matrix from HVC to RA, it starts, here's its initial condition. If we train it with no perturbations, then what you see is that sort of neighboring neurons in the HVC chain, right? So imagine that we're lining up the neurons in HVC according to when they fire. What we find is that the way that they are wired to HVC is very correlated from neuron to neuron. So neurons that fire close together in time also end up largely connecting to the same RA neurons. Whereas if we train the network with perturbation, those correlations are broken. Okay, so we can just quantify that and look at how correlated a neuron firing at time t, how correlated the wiring between a neuron at time t and its connection to an RA neuron depends on the separation in time between the firing of that HVC neuron. And quite clearly, these perturbations sort of kill off that degree of correlation. So why is that good? So here, as we again increase the number of perturbations, the strength of that correlation decays. So where does that come from? I mean, it's not too surprising, I guess, but fact is that when you drive a given neuron with an L-man input, those inputs have a certain time constant, which is not too short. It's, these are NMDA synapses, and so they have kind of long time constants, and so they induce variations that are correlated in time over a couple of 100 milliseconds. And so you end up with networks that are wired up if you don't make any changes that also reflect the time constant of those inputs from L-man fluctuations. As you perturb, you distribute the drive for those song features among different HVC neurons. So you break that correlation. So one way of thinking about that is that in the end, the song is very low dimensional compared to the number of neurons that we have both in HVC and in RA. We only have, I think, 13 muscles to drive. So in our model, we've just taken that to be 2 outputs. And so there are many possible wiring schemes that lead to an equally good song. And so what happens is that the error fairly rapidly gets down pretty low, and then slowly, because of these correlations, continues to evolve toward a point in which the connections between neighboring neurons are very similar. And if we add these perturbations, we can kind of stay away from this very correlated point in weight space, which is also one that's very hard to get out of. If we now need to learn a new song, we're kind of stuck in this potential minimum in which the correlations kind of act against us as we try to do something new. All right, so we just showed that it's not a unique result for this particular kind of perturbation. If neurons shift in time, you get similar results. If they fail in time, you get similar results. The most dramatic results occur if a neuron stops firing for a while, and at the same time, the network continues to undergo LTD. So that's these curves in blue. And so we get the most dramatic sort of advantages from perturbation in the case that happens, but it's pretty robust to decisions about how to induce those perturbations. So this is sort of similar, will be sort of familiar conceptually from machine learning, similar to the idea of dropout. You can train a neural network. It tends to find a very particular solution. You get more robust results if you occasionally sort of switch, drop out neurons in the training regime. And so dropout forces a neural network to learn more robust features that are useful in conjunction with different random subsets. So that sort of explains why our network is more able to learn a new output after having trained it under these conditions. All right, so that was all done under sort of using the same model that Ila introduced years ago. And one issue with that model, sort of conceptually, is that it's not particularly biologically realistic. And so years ago, Michael Faries, when he was in my lab, was sort of disturbed by this point and wanted to see whether it was possible to get reinforcement learning paradigms to work under more realistic learning rules. And so Michael thought hard about how to get this comparable model to work if instead of the learning rule that Ila had introduced, which is sort of a firing rate learning rule, you forced it to work using just STDP, right, which is something we know actually happens in real neurons. And so STDP rule, you allow synaptic changes to occur if spikes occur within a certain time window. You reinforce them if they occur such that the input to output mapping is causal, and then you reduce the strength of the synapse if it's acausal. And what Michael found, and this was not sort of unexpected, is that this kind of doesn't work to reproduce a very well-defined sequence of times. If you want your network to generate firing at a particular time, STDP, as applied generically, say using an additive learning rule, shifts that moment in time. And when people have, of course, seen this before and had even thought about that as potentially an advantage, that spike time independent plasticity can allow you to almost anticipate what's going to happen in the future. But that's not great if you're trying to learn a well-timed sequence. So if you try a multiplicative learning rule, again, you kind of get, you run into trouble that you end up with too many spikes. And so neither of these rules kind of works out of the box to generate a well-timed sequence. So what Michael proposed was that what one needs to do is to modulate your spike time independent learning rule in time according to the error or reward. And so here the idea is that you can evaluate the reward as a function of time and sort of multiply or gate the effect of the learning rule, the spike time independent plasticity over time according to this factor. And so I guess this was an early example of something that people have been talking much more about since then, Wolfram Gussner and others, of sort of tripartite learning, an extra factor that you have to include in the spike time independent plasticity rule in order to get it to work. And so if you do this, you can really beautifully learn very well-timed sequences of spikes. You can learn more complex patterns. And generally, and this is something that Ila noted already also in their original paper, sparse inputs really help you here. So whenever we have a reinforcement learning rule, because we're not doing kind of backpropagation, we're not really allowed to address particular synapses with a particular error, there's a problem of credit assignment. And so clearly, if you have a very sparse wiring such that you have your HVC neurons only fire at one particular time, credit assignment is sort of taken care of by the fact that they are sparse in time. So the fewer spikes that any neuron has to learn to fire, so the number of action potentials in the target train, the better it learns. So that really, and so yeah, here's the performance if you have sparse HVC-like input driving the action potentials. Okay, so you will have noted, everybody, I hope, that okay, these are what we started with Ila's learning rule. We sort of updated it a bit by thinking about adding spike time independent plasticity. But in both cases, what we have done is sort of cheat, right? We've imagined that we have a modulation signal or a credit signal, reward signal, which is present here in the HVC to RA synapses. And that is not true, right? You remember from our picture that we showed here before, that the signal that we believe that carries kind of the critic signal or the evaluation signal, if that is coming through VTA, then that's not in this pathway. It's way over here, right? It's sort of talking instead to the anterior forebrain pathway. And so what's sort of the implication of that? We had thought about this as sort of this random number generator or experimenter, whereas really it's likely that we should be thinking about the anterior forebrain pathway as kind of the critic, right? It's doing the training directly itself. And so Michael, in this paper, hopefully some of you have seen it years ago, thought very hard about how that could work. So what could be going on there? So the LMAN to RA inputs are quite well placed to play that role. The synapses are NMDAs, so they're well placed to generate NMDA dendritic spikes, and so to influence HVC to RA plasticity. It's also true that LMAN fires in two different modes. So during sort of performance mode, it fires with single spikes, whereas when the bird is singing undirected song, when it's kind of more in practice mode, the outputs from LMAN tend to be bursty. And so it's possible that change in mode in LMAN, the bursts are in fact there to heighten the ability to drive STDP. And so it's possible that the transition from directed to undirected song sort of switches the output from LMAN into a way that can either drive or not drive plasticity between HVC and RA. But nonetheless, there's still the question of how one can know, when to switch up or down the wall. And so that suggests that the anterior forebrain pathway can kind of predict the output or act in some sense, at least in maybe short term, as a forward model. And so there've been a number of models over recent years from Michael Fee and Jesse Goldberg, and from Surya and from Bensie's lab that have made that argument that what's going on here is really two-stage learning. There's sort of a short stage learning that's located in the anterior forebrain pathway, and that eventually transforms into longer timescale learning that is happening here at the HVC to RA synapse. And so there's some experimental evidence for that. So as one is training the bird, trying to sing these shifted pitch syllables, if you switch off LMAN, right, after the bird has successfully learned that pitch change, then the pitch change goes away. And so some of the bias that's causing that pitch change is clearly being instantiated by the anterior forebrain pathway itself. Okay, so in general, what's then the right way to think about the role of the anterior forebrain pathway? So one thinks about LMAN, from olden days as sort of a generator of variable firing, and that likely is true, potentially via chaotic dynamics, using models of the type that David has looked at in the past. But it seems very likely that Area X is computing signals that then serve to modulate that output, that can either switch it into a bursty mode or give it sort of temporal shifts that control the output of LMAN. And that ability of X to modulate LMAN's output is presumably trained or modified by signals from VTA. All right, so I'm going to switch gears, how much time we have, and talk about another set of projects, and we'll see how far we get with that. And that is some recent work that we've been doing with Carlos Lois and Tim Gardner's lab. So here, in everything we talked about so far, we sort of took the perspective that HVC is basically acting as the metronome of the song. It's providing the beats that are the scaffold, right, for how to wire up the motor output. And so with Carlos's lab and Tim, what we've been doing is exploring what we can learn about the architecture that allows that to be true. And so Carlos has been doing some viral manipulation of HVC. So in these experiments, we record songs prior to perturbation for a couple of weeks. We inject a tracer to identify HVC, and then inject a virus that perturbs the projection neurons in HVC. So using two different kinds of viruses at the moment, one of which changes firing patterns, one of which silences the neurons. and then record the song post-perturbation and explore the way that the song recovers over time. So what happens to the song? So prior to perturbation, this is very familiar, right? Zebra finch song. After perturbation, one finds that although there are syllable-like events that they break up, that the temporal sequence kind of breaks down, and that over time, the song reestablishes itself. And it can even do that to some degree without auditory experience. So if you prevent the bird from singing, the song still largely recovers, even though it clearly does need some singing to make that happen all the way. So I just wanted to show you, this is sort of a definitely work in progress, but show you some of the things we've been working on with this. So one is, we'd like a way to be able to visualize the song and how it breaks down. And so this is sort of a lovely implementation of UMAP, which is a nonlinear dimensionality reduction procedure. So here, what you're looking at is a sort of a 2D embedding or collapse, right, of the song over time, where each point, it corresponds to a small window of time in the song. And so one of these trajectories is the song as it flows through time. And so we're watching a few good songs, right? So they kind of nicely reproducibly go around this trajectory. And then hopefully at some point, we don't have too many here. Let's see if we can move ahead. The song is perturbed. And now we can watch a few perturbed songs. So for a few days after the injection, the virus takes a while to work. And so the songs remain fairly similar to the pre-perturbed versions. But then the bird stops singing for a little bit. And now I'm gonna zoom ahead. Right, now we start to see some of these perturbed songs. So you see some events that aren't very similar to previous ones. But what's kind of interesting is that these trajectories will kind of jump back to some of the chunks of the original trajectories. So here, right, there's a small fraction of the song that's kind of leapt back onto those original trajectories, sort of suggesting that parts of those sequences that are presumably still embedded within HVC are still accessible. And so you can sort of here beautifully see that there's a whole stretch of song that is sort of recovered over time. All right, so we can use this tool of UMAP. Here we like to see how this sort of flows through time, but we can instead use it to analyze the structure of the syllables themselves. So now just pulling out the pieces that have auditory structure by thresholding on the power. We could use UMAP again to sort of classify those syllables. You see that they very nicely cluster. And now we can look at the syntax of the song in terms of these syllables. And so what Alison's doing here, so this is all work by Alison Duffy, she's looking at, for an individual syllable rendition, how close it is, what is the nearest cluster center to that syllable. And then she also quantifies the distance from that center. And so here we're looking at a transition matrix, so the probability of moving from one syllable to the other. But this representation also captures how variable that syllable is. So the spread of this blob sort of tells you how distributed points are within that cluster. And so here's sort of the grammar essentially of the song prior to perturbation. And then we should see the viral injection happen. Okay, so the song stops for a few days. And now when it comes back, you can see that these syllables are still fairly well defined, but the grammar is totally busted, right? So syllables are transitioning to states that they never did before, but that over time, that grammar, the syntax of the song sort of reestablishes itself so that we return back to a transition matrix very similar to the original one. So we can look at that just in snapshots. So here's the original syllable distribution. Here's the spread of points after the perturbation. And then over time, we see those clusters reestablish themselves. They're not the same exactly as they were before. They come back a little bit different. And here's how the grammar has reestablished itself. And you can see also that it's largely the same, but there is this new transition down here that wasn't there before. All right, so all of this gives us really rich information to start to think about what might be going on sort of under the hood within HVC and possibly beyond HVC in order to allow for this reconstruction. And so here we're talking about work by Rich Pang, Alison Duffy, and one of our new students, David Bell. And so the natural way to start to think about this is, as many people have over a long time in HVC, is that HVC may be essentially a synfry chain. And so groups of neurons, here we think about them as little recurrent subnetworks, communicate with one another in a feed-forward way in order to generate a sequence through HVC. And we also incorporate inhibition, and inhibition is useful because it allows this kind of winner-take-all properties so that only one group of neurons is sort of firing at a time. So inhibition sort of stabilizes and regularizes the chain. And so implementing a model like that, one can test to see whether it does what our real brain does. So if we ablate some fraction of the excitatory neurons, and then try to model recovery, in this case, via synaptic scaling. So one natural thought is that if neurons are not getting the same amount of input that they were before because of these ablated neurons, that homeostatic rules will be at play, and that will sort of scale up the inputs from the remaining networks, the remaining neurons. And so that recovers some aspects of the data that we see. So here's with a small amount of ablation, you see that the network is quite robust to that. And in fact, Carlos's lab have to interfere with quite a large fraction of the neurons before the song breaks down at all. So there is a fair amount of built-in robustness. But if we take it to 30% ablation, then the sequence will in fact be killed off. But now if you apply synaptic scaling, then you can recover the sequence. One interesting finding though is that what we see quite often with the songs is that the syllables slow down. So here what we're looking at is the distribution of the timing, the duration of syllables within the song. So day 159 is prior to the perturbation. Here's the perturbation. Now, if you watch carefully, you see that when these syllables come back, they are quite a bit slower than they were before, and it takes time for them to recover back to their original length. Now, that this kind of chain model can account for that in a kind of natural way. If you think about what drives a given neuron in the sequence to fire, it has some kind of temporal integration window. If it's not getting as many inputs in that temporal integration window, it'll have to integrate for longer in order to propagate the next link in the chain. And so we see this kind of slowing down of the propagation as you drop neurons from the chain. So that kind of makes sense. It's a little more subtle than that though, because if you incorporate inhibition as well, as you drop excitation, you also drop inhibition. And so you're changing that overall balance. And so this data give us a kind of a test bed to study what is the appropriate choice of architecture in terms of what inhibition is really doing here and how structured it needs to be in order to explain our data. And so at a cartoon level, we can capture this behavior, but it's a little bit sensitive to parameter choices. And so we're sort of in the middle of trying to figure out under which combinations of homeostatic and STDP rules do we get the kind of recovery that we see, and what is consistent with the details of the way that the song breaks down and recovers. Okay, so I have a minute or two left, so I just want to very, very quickly just tell you a little bit about a new project that Rich has been working on, Rich Pang, which is the hypothesis that not all of the timing, or that not all of the behavior that we're seeing is necessarily within HVC. And so HVC does get one important input, and that's from area UVA. And UVA is, despite us knowing about that input for a long time, it's still rather mysterious what exactly it's doing. There's very little data, apparently very hard to record from, say, our experimental friends. But the data that we do have comes from this one paper from Michael Fee's lab, where they showed that recording over different regions within UVA, you get outputs that are very stereotyped and song-logged, and that seem to be sort of global. Different regions within UVA seem to have very similar outputs. And so this inspired Rich to ask, what might this be doing what might an input like this do to timing within HVC? And he made the interesting speculation that this might be able to reduce the accumulation of timing errors. So if you have a network that's set up like a chain, then inevitably, as one moves down the chain, the temporal jitter from firing to firing to firing is going to increase in time down the chain. And so there's been interesting work from Tim Gardner's lab proposing sort of detailed structures of an inhibition that could act against that timing jitter. But Rich instead proposed that what we might have is sort of a temporal modulation coming from UVA that might help to control that degree of timing jitter. So we think about HVC as still being a sequence generator, but subject to modulation, temporally varying modulation from UVA that could play that role. And so the idea is that you have a chain network that propagates excitation down the chain, right? These sort of recurrent little subnetworks that are connected in a feed-forward way. And you have some temporally organized signal coming from UVA. He proposes that could be inhibitory. We don't actually really know the sign of the inputs from UVA, which is something that we're hoping that Carlos's lab will help us out with. But it's not enough to just simply give the same input to every neuron in the network. If instead that is also spatially organized, Rich has shown that can serve to kind of to reduce the variability in sequences. So if that modulation was purely spatially uniform, and we start a bunch of sequences with some temporal jitter, that jitter generally will just propagate through the network. So the output will have the same kind of jitter as it started with. If we give it temporally uniform modulation, again, every sequence will be modulated at the same way. But one can figure out how to set up a kind of matched spatiotemporal degree of modulation, where it's the same temporal modulation, but now applied to some subset of the neurons, such that that temporal jitter will actually be controlled. And so the amount of jitter as you move through the network will be reduced by the interaction with these kind of patches of inhibition. And so that, I think it's a cool idea. And so we did our first pass of looking for that in experimental data. And so Alison went back and looked at some of the song recordings that we have and looked at different regions of the song and to see whether there was timing variability from segment to segment that was correlated. And what she found was that variability within one segment was anti-correlated with variability in the next segment, as though there really is a kind of dynamic control of timing that occurs throughout the song. So at least this is sort of the beginnings of some evidence that there really may be kind of long timescale modulation of timing such that the total song ends up less variable than the individual segments. Okay, so going to stop there. So just to summarize what I've shown you is that we see robust sequence generation and maintenance within HVC, and that breakdown and restoration of the song can give clues to understanding the architecture, and we're working on modeling that, and point out a potential role for UVA in maintaining temporal precision. We talked a bit about models of learning, and with the hope that we can move toward realistic learning rules, understand the role of intrinsic variability. We have other projects where we're very excited about trying to understand the origins and the modulation of variability, and also the information content of this dopaminergic feedback. And that's a collaboration with, with Vikram Gadekar and Jesse Goldberg's lab. Okay, I will stop there and just show all my collaborators. Thank you so much to all of these people and to my funding sources for help, and to all of you for being here and for listening to this.